Do you have any questions, class?
Sir, I have question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
Sir, I have question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
Do you have any questions, class?
Sir, I have question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
Do you have any questions, class?
Sir, I have question po.
This pandemic may have changed the way we deliver, but SCCA is dedicated in providing the best quality of education to our students that they deserve. With the combined efforts of our teachers and administrators, we remain steadfast, even during these trying times. We have partnered with advanced learning management systems to make sure that our students can learn at ease at the comfort of their homes. We ensure that we still provide a well-balanced student life through extracurricular activities virtually. To make sure the safety of our visitors and the whole Dominican community, we have strengthened our campus protocols. This 2020 have brought different challenges in the institution, but with the dedication and support of our teachers, staff, administrators, students, and our parents, we are still standing. We will not stop creating new strategies to make the institution more relevant and responsive to the needs of our community and future leaders. This is St. Dominic College of Asia, revolutionizing education beyond borders.
A pleasant afternoon to everyone. Welcome to our first webinar for the month of March. From the School of Health Science Professions, in collaboration with the Department of Pharmaceutical Science, Akhtar Saeed College of Pharmacy in Lahore, Pakistan. I am Janina Dino, former president of the Pharmacy Collegiate Council in St. Dominic College of Asia and national auditor of the Federation of Junior Chapters of the Philippine Pharmacists Association, and I will be your host for today's webinar. Before we start, I would like to recognize the different courses offered by the School of Health Science Professions in St. Dominic College of Asia. We have BS Pharmacy, BS Medical Technology, BS Radiologic Technology, BS Nursing, BS Biology, and BS Physical Therapy. Here is the flow of our program for today. First will be the doxology, followed by the Philippine National Anthem, welcoming address delivered by Professor Roland Sardan, introduction to the guest speaker by Ms. Ira Nicole Tibayan, our guest speaker, Dr. Sulaman Yakub, the question and answer portion, followed by the reaction of Professor Anthony R. Marine, and then the closing remarks of Professor Yurek Araneta, and the awarding of certificate, followed by the pharmacy hymn and SDCA hymn. May we request all student attendees to comment their names and program for their attendance. Now let us start our program with a prayer, followed by the Philippine National Anthem. At this point in time, to open and welcome today's webinar, let us all welcome the Program Chair of Physical Therapy Program, Professor Roland Sardan.
Thank you very much, Ms. Dino. So, in behalf of our Vice President for Academic Affairs, Dr. Marinelli T. Roa, and our School Dean, Dr. Joel G. Matamis, I would like to welcome everyone to our first webinar sponsored by the School of Health Science Professions through the efforts of the College of Pharmacy, entitled The Role of Medicinal Chemistry in Drug Design. As early as now, we would like to thank our esteemed speaker for today, Assistant Professor Dr. Sulaiman Yaqub from the Department of Pharmaceutical Science of the Akar Saed College of Pharmacy in Lahore, Pakistan. Thank you very much for accepting our invitation and gracing our webinar for this afternoon. It is our hope that our students, especially the students of the College of Pharmacy, will be able to grasp different concepts of the importance of medicinal chemistry in the different practices and professions under the School of Health Science Professions. So, in behalf of the administration and the other program chairs of the School of Health Science Professions, we would like to welcome you to our today's webinar, and we hope that you will get important learnings and have a good day ahead of everyone. Thank you very much.
Thank you very much, Professor Sardan, for that wonderful opening remarks. At this point in time, to formally introduce our speaker for this webinar, may I call on Ms. Ira Nicole Tibayan, third year BS Pharmacy student and third year representative of the Pharmacy Collegiate Council.
Today's guest speaker is a young researcher and scholar from Lahore, Pakistan. He has graduated in science with chemistry, botany, and zoology from University of the Punjab, a well-renowned university of Pakistan. He pursued his career as chemist in a multinational chemical industry. Later on, he qualified PharmD Doctor of Pharmacy, MPhil Pharmaceutical Chemistry, Scholar from Punjab University College of Pharmacy. University of the Punjab, Lahore, which is ranked among 251 to 300 global schools of pharmacy by QS ranking in 2021. He has diverse experience in pharmaceutical industry with hands-on training on various aspects dealing with analysis and formulation of drugs. Based on his vast industrial background, he joined academia and opted for pharmaceutical chemistry as his major strength. His field expertise relates with medicinal chemistry, drug stability, and pharmaceutical analysis. Currently, he is working as assistant professor in pharmaceutical chemistry in Akhtar Saeed College of Pharmacy, a well-acknowledged pharmacy college of Pakistan. Please welcome our guest speaker for today, Dr. Sulaiman Yaqub.
Thank you very much, Miss Arya Nicole, for such a nice introduction. Thank you very much, Professor Ronald, for welcoming overall, and special thanks to Dr. Anthony for inviting me to this webinar, and the thanks to St. Dominic College of Asia. So before proceeding to the lecture, I would like to share my thoughts towards the college. I'm very much inspired by that the ads were broadcasting on the live streaming, introducing your school. I'm talking especially to the students that you're very lucky that you're learning in such a nice capacity and infrastructure. And I wish you best of luck. I hope you will learn something new from me, definitely. So I guess we should start with the presentation. uh, Please share the slides on the screen. Thank you very much. So our today's topic is the role of medicinal chemistry in drug design. It's very interesting to understand the medicinal chemistry as it is the basis of all the pharmacy sciences, especially related with the drug designs. Please, next slide. First of all, we must know about the medicinal chemistry. I hope the students who are in final year, they are studying the medicinal chemistry as per your national curriculum of pharmacy. But those students who are junior or who belong to other disciplines, they must know about the medicinal chemistry. It's the combination of various chemistries and other subjects that are related to the drugs. We can say it is the highly interdisciplinary science as it relates with the various disciplines, especially organic chemistry and pharmacology. These two are the major sciences that are related with the drug design and the aspects of drugs and the functioning of the drug in the body in special relation to their structure and their overall function. Other sciences that we can relate with the medicinal chemistry that we must include are biochemistry, the basic knowledge of, or you can say the background of pharmacology is biochemistry. And similarly, toxicology, pharmacognosy, molecular biology, genomics, proteomics, computational chemistry, physical chemistry, statistics. We combine all of these sciences together to make new drug molecules, to refine the pre-existing molecules, to eliminate the adverse effects and many more functions that we can resolve with the help of medicinal chemistry. Next slide, please. So, as our today's topic is particularly related with the drug design, so I must have the emphasis on drug design that uh, why we need to design a drug. Definitely, new ailments and diseases are found day by day. So we must have some new drug molecules to tackle all these problems and ailments. So to find new drug molecules, we need to identify the therapeutic usefulness of a compound against a particular target, drug target. And in this process, we generally addresses those biological targets that play some important role in the mechanism of disease. As a disease condition is there, there must be some background of biochemical pathways, biochemical signaling, and other various aspects that are the alteration in the normal physiology of the body. So we address those biological targets that can be modified by certain therapeutic agents. Sorry. So those therapeutic agents are then related to the biological target, and their usefulness are identified whether they are useful in modifying the particular biological targets or they are not. If they are not, then we must try the new therapeutic agents. So it involves few steps. I can say the initially we need to identify the candidates of drugs as the compounds or the molecules that have potential to be a drug. And these drugs can be obtained naturally from the various sources, as we will discuss later on, or they can be synthesized in the laboratory. And after their synthesis and their identification, we need to characterize them. We need to find them. We need to find the properties of them, their behavior within the body, and their behavior in the diseased condition as well as in normal physiological conditions. Then we screen those molecules, and after the screening, we conclude some smaller fraction of molecules, and to that smaller fraction, we develop assay of therapeutic efficacy, then pharmacokinetic studies, then other studies as the toxicological studies. And after these studies, extensive studies, we conclude one or two drug molecules that are capable, safely capable of exerting some therapeutic effect on the body. Next slide, please. So here is an overview of the drug discovery process. We will discuss this slide at the end, but initially I have mentioned it here so that we must know the overall circle of the drug discovery. It's a very complicated process. We cannot simply understand it by just reading it from the book. Technically, it involves a lot of screening and different and complicated processes. Here I try to simplify it as much as I can so that the students, final year students, especially the other students, they could have the idea of how a drug is optimized, how a drug is discovered and developed before it came to the market. So we will discuss this slide in the end. Next slide, please. So, among the steps that I have mentioned here, the medicinal chemistry helps in the discovery and characterization of potential drug compounds, which we call hits or lead compounds. These are the compounds. Technically, the drug development is a very long process. It involves both the medicinal chemistry as well as pharmaceutics, pharmaceutical technology, and other pharmacy sciences as well. So the first step is the most critical step, and here is the role of medicinal chemistry, that it helps us in discovering and characterizing the potential drug compound. Without medicinal chemistry, we can never find a drug candidate that is suitable for a particular biological target. For this, we need to identify the hits or lead compounds, and we make thousands of variants of these compounds. Among thousands of compounds, we screen the best suited lead compound, and we use that lead compound to the therapeutic purposes. Next slide, please. Next slide, please. So here are the steps of drug development. Drug development is a very complicated process. Here I have summarized some aspects of the process. Initially, among which we need to find the biological target, which we are going to address, and then the potential drug candidate that is going to modify that drug target. And from that, we find lead compound, and the lead compound is the best suited compound that interact bestly with the biological target. And then we optimize the lead compound. We remove the toxicological aspects of the compound, and we modify the functioning to enhance its various aspects. And after the optimization of lead compound, it is being formulated and packaged. Its formulation studies are done. Then its clinical studies, preclinical and clinical studies are done, and the finally drug is ready to submit for approval. And if its data support the drug, then drug is approved in the local country regulatory body. Every country has its own regulatory body. Definitely, there must be some regulatory body in Philippines where anybody can submit the data and approve the drug to sale in the local market. Similarly, we have DRAP here, Drug Regulatory Authority of Pakistan, and you would be well familiar of FDA, etc. Next slide, please. So here is the process diagram of drug discovery and development. We call it the drug discovery and development pipeline. So it starts with the target identification. We need to identify the target, particular drug target. That target will be addressed with the help of some potential drug candidate. After the target identification, we validate the target, and the target is validated whether the target we are addressing is specifically involved in that disease condition which we are addressing. And after the target identification and validation, we find heat compound or lead compound that I have told you. So that heat and or lead compound will then be optimized, and after optimization, it will be sent to preclinical trial. But before preclinical trials and during optimization, we study the pharmacokinetics, the general pharmacology, and toxicological studies of the compound, which enable us to understand the probable effects of that drug in vivo. Before in vivo studies, during optimization, again, we have in vitro studies as well. After preclinical trials and the successes, we shift the drug to clinical trials. And after clinical trials, it is approved. Next slide, please. So, in all these processes, medicinal chemistry is related with the drug discovery. How we are going to discover the drug, mean the potential candidate, which we are going to use. We need to find that potential candidate. So for this, we have a rational drug design, which is a systematic approach of target-based identification of drugs. It means we identify drugs specifically on their action on particular drug targets, biological targets. So it is the very conventional method of target-based drug design, and the drugs from natural and synthetic sources are obtained, and then they are studied for drug discovery. Natural sources include plants, animals, microbial and marine sources, mineral sources. We are right now we are obtaining drugs from all these sources, and those drugs. There is another source we call semi-synthetic. Semi-synthetic drugs are technically the naturally origin drug, but we modify their natural structure with respect to our particular biological target. And finally, the synthetic sources, it means we identify some receptor, and then we find the drug target after docking, a long and complicated docking procedure, from which we find a suitable drug candidate. So the different aspects or different types with which we can identify a drug is the different designs of drug formulation. One is the ligand-based drug design. It means we identify a drug based on the ligand. We call it a receptor fit model. It means those substances, those drug candidates that fit with the ligand suitably, and they are capable of modifying the receptor and initiating some response from that receptor. Secondly, are the structure-based drug design. Structure-based drug design, we specifically note the structure of a drug molecule, and the probabilities of the structure of that structure for binding and initiating some response are calculated on the computers or other techniques. The third one are specifically computer-aided drug design, in which we design molecules. We model the molecules as per our requirements. We modify the molecules as we need them to be. Next slide, please. Next slide, please. Okay, so drug discovery. Drug discovery, again, as I told you, the rational drug design. Here are the components of a rational drug design. Previously, we discussed various designing methods, and here is a general overview of the rational drug design. First of all, the target is identified. Then the target is validated, and various methods we use for validation are high throughput screening or virtual high throughput screening, then lead optimization, lead identification and optimization, and the drug sources, etc. This is the summary of all the previous slides I have told you. Please, next slide. So the first step is the drug target. How we can identify the drug target. For this purpose, we need to understand the mechanism of a disease. We need to find the exact signaling pathway that is carrying out that disease. So, after identifying the metabolic or signaling pathway, we can find out the potential drug target. Potential drug target is the key molecule that is involved in metabolic and signaling pathway of any disease or infection, or even the survival of microbial pathogen. If we focus only on disease, we can never cure that disease. So we need to find out the even the survival mechanism of microbial agents so that we can kill them and cure the infection. So these are the then we find the interaction of proposed drug candidate with biological target. It means after selecting the particular drug target, we need to find those drugs molecules that can interact and modify the drug target. How? They can inhibit the disease by blocking the key molecule in disease pathway. As we already know, the molecule involved in the disease pathway, if we block that molecule, we will definitely cure that disease. And the second option is, as there are always most of the mechanism occurring in the body are antagonistically. If one thing is increasing, the other thing is decreasing, so that the level is on average, the level remain on average. So we enhance the normal metabolic pathway by promoting specific molecules. So these two are the strategies of drug receptor interactions. Next slide, please. So the drug targets, they may be of two types. One are the established drug targets. Established targets mean those drug targets whose mechanism is fully established and understood, mean we have understood those and published the functions of those receptors of those drug target molecules in the normal physiology. We know each and every aspect of that molecule, where it comes from, how it behaves, and what will be the fate of that molecule. When we know such information about the target, we call that target as established target. So established target helps us in finding the chemical feasibility for developing small molecular therapeutic agents against that target. It is always helpful to find the small molecular therapeutic agents because it is very convenient to modify them and to use them. The second category is the new target. New target does not mean totally new target. It means those those protein molecules that we have studied, that are established, but they are not well established. We have discovered them. We know their general function, but their specific function is not known. So before proceeding any further, we need to establish these new targets. So it means established targets are always much easier as compared to new targets. So generally they involve two types of proteins receptors. One is the G protein coupled receptor. You all are familiar with the G protein coupled receptors, you have studied in pharmacology. And the second one are protein kinase. Both of these proteins help in mediating some response from the cells. Next slide, please. After identifying the target, we need to validate that target. Process of gathering functional information of target, we call it as target validation. It means we verify the target molecule is directly involved in the disease process or not. So this validation is very necessary. There sometimes there may be some misunderstanding that can lead to wastage of time and money. So we first need to validate the target, that we are addressing the right molecule. After validation, we pass the drug molecules to high throughput screening. Large libraries of compounds are tested for their abilities to interact and modify target. For example, if the target is a novel G protein coupled receptor, then the drug is tested for its ability to either stimulate or inhibit the receptor. We all know that G protein coupled receptor work in both manners by different drugs stimulate the G protein coupled receptor, and many drugs they inhibit the receptor and initiate some response. The other example is if the target is protein kinase, then the drug is tested to inhibit that target. Next slide, please. So there is one very important process that we know as cross screening. Cross screening means we have a drug molecule that is affecting a particular drug target, but what if that particular molecule affects several other targets as well? So, to identify the selectivity of the drug candidate, we need to find the its activity, its interference with other targets as well. So, the interaction of drugs other than specified biological target is identified, and after its identification, we establish the probable toxicity of the drug candidate. How? If a probable drug molecule interacts with so many unrelated drug targets, biological targets, so it means its off-target toxicity would be very greater. That would be a challenge later on. So after cross-screening, we eliminate those candidates that interact with unrelated biological targets. We only focus on those drug candidates that interact specifically and selectively to the specified biological target. For this purpose, we use high throughput screening, and there are various methods of high throughput screening. We use virtual high throughput screening as well, in which the screening is done using computer generated models and the docking virtual libraries to a target. We have a lot of variants of a single drug candidate, and all of those thousands of compounds in which are which have small differences in their structure, and we dock them to their target and observe their results. And we have computational tools in which we don't need to physically interact individual molecule thousand times in a different way. By computational tools and virtual high throughput screening, we can do this on computer. We call it in silico evaluation of drug candidate. Structure of the drug molecule that can specifically interact with biomolecule can be identified with the help of computational tools. And after its identification, that molecule is chosen as hit compound or lead compound. Next slide, please. So it is a very interesting, you can say, the part of molecule that is responsible for particular biological and pharmacological response. We call that part of molecule as pharmacophore. If a substance, if a drug candidate have a pharmacophore, it must have some biological activity, some pharmacological action that we can use for therapeutic purposes. And if a drug molecule does not have any pharmacophore, it will not be used for therapeutic purposes because it cannot initiate some pharmacological response. So to identify the pharmacophore is the major function of high throughput screening. We identify pharmacophore, and depending upon the pharmacophore, additional functional groups are attached with the drug molecule to improve its various properties, and the best lead compound is then identified and optimized for drug development. Next slide, please. So the identification of lead or hit compound, as I mentioned, undergo a very hard and tough screening, after which we find out the lead compound or hit compound from the compound library. That lead compound is undergo through the high throughput screening, in vitro activities, in vivo activities, then pharmacokinetic studies, toxicity studies, and best suited compound is identified as lead. Rest of the compounds we call as backup, because if the lead fail, we find some other lead compound from the backup. We do not waste our backup because it's the very costly and the fruit of a very hard effort. Next slide, please. So the processes of in vivo, in vitro studies, pharmacokinetic studies, and other studies, we finally find out the lead compound. The process of these studies, we call it as lead optimization. Here is a very simple diagram to understand that we have a lot of different compounds that are potential drug candidates. Those compounds are filtered through a sieve, and we have many filters in that sieve. We identify pharmacophore, we have various formulas, we find docking, we find protein receptor interaction, then we find in vivo, in vitro studies, pharmacokinetic studies, its distribution in the body, and many other aspects. And after all these studies and tests, the best compound that pass through all these sieves and we find that compound useful, we call it a lead compound. So this diagram shows two design methods. One is the ligand based drug design and the other one is structure based drug design. Ligand based drug design, in ligand based drug design, we identify the specific ligand and the interaction of that ligand with the drug molecule or the receptor molecule. Receptor and drug interaction are most important in ligand based drug design. While structure based drug design, we identify the structure of a compound and structure of the possible receptor or the biological target, and then we merge. We call the merging as docking. Docking means the interaction of drug molecule and the target molecule with each other. And if we find the expected or the sufficient result, we pass that compound and to transfer it to lead compound. Then the after lead optimization, we finally get a compound that we can use for preclinical and post clinical or the clinical studies of the drug molecule. Mind you, among thousands of compound, we only have 10% of the compound that pass lead test. So when we get lead, we say we have 10% of the compound, and after optimization, we only get about 10 to 20 drug molecules that are optimized. And after pre-formulation and formulation studies and other pharmacokinetic and other studies, we only get one or two molecules among thousands of molecules. So it is a very costly and complicated process. Next slide, please. This is the lead optimization that I have mentioned. We modify lead compound by their chemical alteration on the basis of structure activity relationship. It means when we find the lead compound, we modify the structure of that lead compound so that we may get the optimum result from that lead compound. We use the SAR data, structure activity relationship data, as well as the stability data for that compound. And this lead optimization improves biological and biochemical properties, binding properties, and they also improve the interaction of drug molecule with the receptor or target molecule, as well as pharmacokinetic, its proper distribution, its absorption, distribution, metabolism, and elimination, ADME, that we call the pharmacokinetic. We improve the pharmacokinetic of the lead compound by optimizing the structure and stability data. Then finally, the reactivity and stability during metabolic degradation is also addressed. So hence the compound is optimized and ready for preclinical trials. Next slide, please. So here is the summary of all that I have told you. We started from the left lower corner, database construction and management. Initially, we construct a database and we manage the database. We use computer-aided drug designs, and from that drug design, we identify the drugs and targets, and then we dock them with each other. We watch, we study the interaction of drug and the receptors, and from that we virtually screen the drug compound and we find the lead compound. And after the lead compound is obtained, we modify and optimize the lead compound, and that optimized and lead compound is then further transferred to the pharmacological and toxicological studies. And after passing these toxicological and pharmacokinetic studies, the drug is finally ready for preclinical and clinical trials. Mind you, before preclinical trials, we estimate the possible pharmacology by in vitro analysis. We devise certain mechanisms, we devise very extensive techniques to identify the drug receptor interaction and possible effect of a drug on the receptor. We can even quantify that reaction, and we can say even a drug after in vitro studies that the drug is stimulating a receptor how much, and the drug is how much blocking or inhibiting the receptor. So hence we have preliminary data that helps in establishing preclinical trials and animal trials. After passing the drug in animals, we apply the drug to human, and phase 1, phase 2, phase 3, phase 4, and so on. The clinical trials are done, and finally the drug is sent for approval to the local governing bodies or the regulatory bodies. The regulatory body, after obtaining all this data and analyzing the data, establishing the safety profile of the drug, it approves the drug to sell in the local market. I hope you must have find this lecture very interesting. If you have any question, feel free to ask me. Thank you very much. That's it from me. Thank you very much.
Thank you very much, Dr. Yakub, for that very informative and well delivered discussion. We shall now move on to the question and answer portion. Should you have any questions for our speaker, please don't hesitate to type them in our comment box down below, and we will be reading them for you. So, first question we have, um, how exactly do pharmacists or chemists modify lead compounds to become a medicine that has best efficacy and least side effects?
I have mentioned that it is the structure activity relationship. We have structure activity relationship data. Now we have established studies for various pharmacophores. Pharmacophores are technically various functional groups on the drug molecules that interact with the receptor and exert some pharmacological action or even some toxicological affection. For example, in general, the amino group are toxic in nature. So when a drug molecule have certain amino group in specific environment, definitely it will cause toxicity. So we remove the amino group from that drug compound and we test it again. And if the toxicity is decreased, it means our task is accomplished. And if it's not, then we modify other structure. We generally shift functional groups from one part of the molecule to the other part, or even we change the functional group. But mind you, before doing all this, we must keep in mind the pharmacokinetic, because if we change the functional group, that will definitely alter the polarity and solubility of the drug molecule. Hence, the distribution of the drug molecule in different tissue is also affected.
Thank you very much. Um, Next question we have: what roles do medicinal chemists play in discovering and developing new drugs?
Actually, this is all about the role of a medicinal chemist or the pharmacist expert in medicinal chemistry to develop new drug. Because a new drug molecule, we need an extensive knowledge of structure of the drug molecule and the possible properties, physical properties of that drug molecule due to its structure. So when we know the structure, and definitely only chemists know the structure of a drug molecule, we can predict each and every aspect of the drug molecule. Here we are capable of even predicting the possible pharmacological and toxicological effects by just looking at the structure of drug molecule.
Right. Next question: what is the most challenging part in drug discovery?
The most challenging part is the identification of lead compound. When we find lead compound, it is far easier to modify the lead compound. But it is just like to find the pin in the heap of a straw. It's very difficult to find single compound they capable of interacting with the receptor molecule. So if we find the lead compound, the rest is quite easy. It's not easier, but compared to the beginning, it become easier.
All right. For the last question, we have: in today's global crisis of COVID-19 pandemic, how valuable
Please,
is
I could not listen. You please repeat. Please
in
repeat.
today's am I audible?
Yes,
In
please.
today's global crisis of COVID-19 pandemic, how valuable is starting point for the identification of the lead compounds for the identification of effective treatments?
It's very interesting. So, first of all, you need to have the established study of the virus, COVID virus. When you know about the virus, each and every aspect, then you start making the drug compound that can stop that virus. It means, first of all, we need to identify the structure of the virus, I mean, the chemical components in the virus. Second thing, we must understand the mechanism of infection, how this COVID-19 SARS virus affect human being. I mean, how it enters. We know it enters through the nose and the mouth, and it enters into the lung. What happened when it entered into the lung? How it cause infection? How it affects? A recent study indicates. Initially we know that it affects the lungs, and the patient with COVID-19 infection, their lungs are destroyed and they died of hypoxia. But later on, we find that this COVID-19 virus, it's a recent study, that it enters into the heart, and the heart enzymes and the heart mediators are increased. Many patients, even in my surrounding, I know a couple of patients that are died due to cardiac arrest after COVID infection. So here things are not clear right now. First, we need to identify the exact mechanism of the virus, how it is causing infection. That I have mentioned, that we must know the disease mechanism. When we know the mechanism, we can find any molecule in between that will be the key molecule, and we stop that molecule to cure the disease and to break the infection. So that's how a medicinal chemist can input in this global scenario.
Okay, thank you very much, Dr. Yakub, for entertaining all our questions for today. So, at this point in time, I would like to give the floor to the program chair of BS Pharmacy, Professor Anthony R. Marine, for his reaction.
Good afternoon. Am I audible? Okay. Thank you, Miss Janina Dinu. You did a great job as moderator for this international webinar. Good afternoon, everyone. To the President, Dr. Gregorio Andama Jr., Vice President for Academic Affairs, Dr. Mary Nelly Tiroa, the Dean of the School of Health Science Professions, Dr. Joel G. Matamis, faculty and staff, good afternoon again. Salam alaikum to you, Dr. Salaman Yaqub.
Walaikum assalam.
It is our honor and pleasure to be our speaker today's webinar.
Thank you very much.
The
It's
School of
very
Health Science Profession and thank you. The School of Health Science Professions and the Department of Pharmacy are grateful for your time and support in this event. This webinar will help our students and the listeners to know the importance and the role of medicinal chemistry in drug design. In medicinal chemistry, the chemist or the pharmacist attempts to design and synthesize a medicine or a pharmaceutical agent which will benefit humanity, such as a compound called a drug. Medicinal chemistry, it concerns the discovery, the development, identification, and interpretation of mode of action of biological active compound at the molecular level. As stated by Dr. Suleiman Yaqub, that it involves different interdisciplinary science, such as biochemistry, pharmacology, molecular biology, and so on and so forth. Dr. Yaqub also emphasizes that medicinal chemistry covers the three stages. Discovery step, which are involving choice of therapeutic agent or therapeutic target, such as receptor, enzyme, and target group. Also cellular or in vivo model. Number two is identification or discovery and production of new active substance interacting with selected target and compounds. We call it the lead compound. And number three is optimization, which deals with the improvement of lead compound. The optimization process takes primarily into account the increase in potency, selectivity, and toxicity development stage, whose purpose is continuation of improvement of pharmacokinetic properties. Generally speaking, in medicinal chemistry, we can identify the following stages in drug discovery, design, and development. Drug discovery, it means finding a lead, choose a disease, choose a drug target, identify bioassay, find a lead compound, isolate and purify the lead compound if necessary, determine the structure of the lead compound if necessary. And for the drug design, as stated by Dr. Suleiman Yaqub, that it's used to identify the structure activity relationship, or we call it SARS, identify the pharmacophore, improve the target interactions, known as pharmacodynamics, what the drug does to our body, and improve pharmacokinetic properties. It means what the body does to drug. And today, to our students and to our listeners, I think they learned something, and I will grab the opportunity to thank also the people behind this event, Ma'am Stephanie Palpalatok, Ma'am Jem Cruz, Ms. Nicole Tibayan, Ms. Siren Pavo, and Ms. Camille Iris Simbulan, Dr. Iman Pacao, the faculty and staff. Thank you all for your support. Again, on behalf of the St. Dominic College of Asia and the School of Health Science Professions, we would like to thank you, Dr. Sulaiman Yakub, for the informative discussion in terms of the role of medicinal chemistry in drug design. Maasalam for now, and salam alaikum to you.
Walaikum assalam. Thank you very much. I am so obliged by St. Dominic College of Asia, and it's my best webinar here. Thank you very much, all of you.
Thank you very much.
Thank you for your very comprehensive and well-versed reaction, Sir Marine. And now let us present the Certificate of Appreciation to our guest speaker. Citation reads, this Certificate of Appreciation is presented to Dr. Sulaiman Yakub, Assistant Professor, Department of Pharmaceutical Chemistry, Akhtar Saeed College of Pharmacy, Lahore, Pakistan, for sharing his expertise and valuable knowledge as a speaker during the pharmacy webinar with a presentation entitled Role of Medicinal Chemistry in Drug Design as part of Virtual Pharmacy Education 2021, delivered via Facebook Live given this 20th day of March 2021 at St. Dominic College of Asia, Emilio Aguinaldo Highway, City of Bacoor, Cavite, Philippines. Signed, Professor Anthony R. Marin, RPH, MS Pharm, Program Chair of College of Pharmacy, and Dr. Joel G. Matamis, the Dean of School of Health Science Professions. Congratulations and thank you very much.
Thank you very much.
At this point, may I call on Professor Yurek Araneta, the Program Chair of BS Radiologic Technology Program, for his closing remarks.
Thank you, Ms. Dino. So first, I would like again to recognize the support and presence of our Dean of SHSP, Dr. Joel Matamis, our Vice President for External Linkages, Dr. Nilda Balsicas, and to our ever-supportive Vice President for Academic Affairs, Dr. Mary Nelly Roa. Also, to the Program Head of College of Pharmacy, Professor Anthony Marin, for spearheading this health information webinar entitled Role of Medicinal Chemistry in Drug Design. But of course, most importantly, it was our honor and liking to have Dr. Sulaiman Yakub for sharing his time and expertise to us all. So truly, it is an informative day to us. So today, March 20 of 2021, the community of School of Health Science Profession in St. Dominic College of Asia had witnessed another interesting health information webinar related to all of our chosen professions, which is truly an insightful and flavorful study on the role of chemistry in drug design that is timely relevant to what is actually happening in the world right now due to this pandemic period. Having that, now more than ever, we must engage ourselves with knowledge related to medicinal drugs, which is obviously beneficial to all mankind. So to formally close this webinar, let me leave you with a quote by Hippocrates stating, "Wherever the arts of medicine is loved, there is also a love for humanity." With that, again, thank you to our participants and viewers who had led their time with us, and expect for a more substantive webinars like this to come. Cheers and have a great day, everyone.
Thank you very much, Professor Araneta. So please don't forget to sign for the attendance link flashed in your screens right now for your e-certificate. Indeed, discovering drugs is a very tedious process, but it only takes enough patience, perseverance, and dedication in order to be successful in creating and developing such. This has been your host, Janina Dino. Thank you very much, and may God bless us all. Let us now hear the pharmacy hymn to be followed by the SDCA hymn to properly end this program.
All the ages gone by, to the years that are yet to be, pharmacy thou stand ever glorious, the mission of integrity, the hand that shaped our past, the mind that made the last, shall always be our guardian and brightest hope of all men. With voices from our grateful hearts, we hail pharmacy. Through all the ages gone by, to the years that are yet to be, pharmacy thou stand ever glorious, the mission of integrity, the hand that shaped our past, the mind that made the last, shall always be our guardian and brightest hope of all men. With voices from our grateful hearts, we hail pharmacy. With voices from our grateful hearts, we hail pharmacy.
Swastika we sing, with joyful hearts we bring, noble mission and vision that everyone holds on, culture of excellence and goals to reach our dreams, with unity and flavor we cherish forevermore. Alma Mater, you guide us towards the truth that every one of us has to go for.